Welcome to Qlucore Omics Explorer. In this tutorial, we will show you how to import a VST normalized dataset using the wizard, and how to explore the data and to perform statistical analysis. We assume that you're familiar with the basic concepts of Qlucore Omics Explorer. When using the Deseq2 package in R, the data output can be a normalized data matrix after variance stabilizing transformation, VST. This file can be imported with the wizard in Qlucore Omics Explorer. With the wizard, you can import normalized data as well as annotations connected to your dataset. There is a more extensive video tutorial covering the wizard. This is the VST file from the Deseq2 package that we will import. It is a public dataset with samples from colorectal cancer patients. Let's start. We open the file with the Open with Wizard from the File menu. We select the VST file. The VST file is typically exported from R in a tab-separated TXT file or CSV file format. We step through the wizard using the Next button and follow the instructions. The first question, is this a raw count matrix? This is a VST normalized data, so the answer is no. You then press Next and step through the wizard. We answer questions and mark the parts so that all the data, including variable annotations and sample annotations, is imported. You now have a VST data matrix loaded. Remember to save the data using Save As in the File menu and save the data in the Qlucore GEData format. This will make it faster to open the dataset the next time. In this dataset, we have colorectal cancer patients. We will be using a total of 54 samples. From each patient, we have 3 samples: normal colon, primary tumor, and liver metastatic samples. If you don't have the sample annotation information with the VST data to begin with, you can then simply add sample annotations with Import Sample Annotations in Qlucore. The variables are identified by Ensemble Gene ID. You can, as example, use Biomart to obtain the corresponding gene symbols. These can be imported with Import Variable Annotations in Qlucore. We will first look at the data to see if clusters can be identified. To do so, we open the Statistics window. In the Statistics window, under the Variance tab, you can filter your data to find the highest projection score, which will help you to identify possible clusters in the dataset independent of any annotations. The PCA plot, but also other plots and methods available in Qlucore, like t-SNE plot, hierarchical cluster, k-means plus calculation, can help you to find clusters in your data. Now let us further inspect the plot. As we have some sample annotations, let's see how they correspond to the clusters. We color according to the annotation primary, metastatic, and normal, with the 3 groups primary colorectal cancer, normal colon, and colorectal cancer metastatic liver. We can see the 3 groups where the metastatic samples separate well apart from 2 samples. The other 2 groups also cluster well together in the PCA plot. Since we have annotations that we can use, we can perform statistical analysis directly. One established way to find discriminating variables in VST data is to use the lemma function that is implemented in R. This can also be used directly from the Extended Statistics in Omics Explorer. Please see our manual for installation of R in the packages needed. For more reading about lemma in VST, you can, for example, study a comparison of methods for differential expression analysis of RNA-seq data by Sonesson and De Lorenzi. We now remove the variance filter to bring back all data. In the Variables tab, we specify that we would also like to see the gene name in the Q value column. For example, we may want to look for discriminating variables between normal colon and primary colorectal cancer. We can uncheck the group colorectal cancer metastatic liver. In the statistics window, we select Lemma T2B1, the annotation primary, metastatic, and normal, and press the Run button. The lemma function in R is now called, and after a while we get the done feedback. We can now use the slider to filter out the best discriminating variables. You can see how the samples cluster in two groups. In the Variables tab, we can order them with the gene with the lowest Q value on top. You can now copy the data into a variable list and export the variable list to file. Find more video tutorials on our website. If you have any questions, please don't hesitate to contact us. Thank you for watching, and good luck with your analysis.